determine the underlying genetic components? And can we even hone down to the genes responsible, gene or genes responsible for governing some aspects of these complex traits, okay? So that's what today is all about. It's all about the power of genetic crossing, which is, of course, super exciting. Now, people, say, oh, okay, quantitative trait loci, QTLs, mapping QTLs. I just want to say, for those of you who may have missed it, this came up in the question and answer period, QTLs are loci that are associated with variation in quantitative or complex traits, okay? They're the underlying genetic loci. And instead of just saying quantitative trait loci over and over again, we shorten it to QTLs. So today is about mapping QTLs. Mapping QTLs associated with, with complex traits in plants, all right? What are some of the approaches we have to doing that? Now, I have to say that, today you're going to learn about some of these scientific approaches, and it's about learning to think like a scientist, right? There are other alternatives which are in more and more wide use now, such as things like genome-wide association studies, right? I am not going to be talking about that today, because these alternatives, the reason why they're in wide use is because, oh my gosh, we have so many genomes getting sequenced more and more and more and more rapidly every day. Those types of studies rely on certain assumptions, which include things like, they're basically association mapping that assume things like random mating in populations, right? That basically try and take advantage of the natural variation contained in our sequences, us and other, organisms, and therefore do not require crosses. But because they do not require crosses, they are subject to a whole wide variety of drawbacks, which include some of the assumptions that they were making, like a random mating population. That is obviously something that's generally speaking violated. And what that means is that these genome-wide association studies are subject to high false positive rates, okay? So they are increasing in popularity because of the type and nature of the data that we have, but on the other hand, it's, I want you to learn about some of the studies that can actually hone in on which genes are responsible. Okay, and it also highlights the importance of developing model systems. The importance of developing model systems that we can manipulate in the laboratory in order to better understand the genetic components underlying complex traits, okay? So this is the question we're focused on today. How do we determine which genes are involved in controlling a particular quantitative trait that we might be interested in, okay? And I'm going to break that down for you today. Okay, so as I said, the genes that control variation in quantitative traits are known as quantitative trait loci. These QTLs generally have allelic variants that make usually small quantitative contributions to the phenotype. A quantitative effect is something that can be attributed to two or more important genes, could be anywhere from just a few to hundreds, okay? Now, QTL mapping is what people do to try and determine where their location might be in the genome. And there has been lots of pioneering work in QTL mapping. In fact, it was first done on crop plants for reasons that you will see in a minute, because it involves lots of crossing of isogenic lines, right? But now it's broadly applied to many different model organisms. So the fundamental idea, though, behind QTL mapping is that you can identify the location of a QTL in the genome by using marker loci that are linked to the QTL, right? So incorporated in this approach is also statistical methods that allow us to identify, once we have collected the data, where in the genome our QTLs might reside, okay? All right, so let's talk about this in more detail. How actually is this done? And I'm going to talk about this step by step, all right? I want you to understand what this whole approach is about and what it's possible to do in science, all right? Okay, so the first step is to make a cross between two inbred strains that differ, and this is really the key part, right? It's not just, oh, I have two inbred strains of tomato. Maybe I should cross them and find some QTLs. No, you don't want to do that. Why wouldn't you want to do that? The reason is because you want to carefully select your inbred strains, one, to have as much difference in your quantitative trait as possible, okay? And this is very important because you are limited to the genetic diversity that is present in the parental strains that you cross, right? If your parental strains don't have alleles associated with that trait, or if they have alleles that only have, very small effects, your chances of success are going to be very small, okay? So you want to make sure that you cross two strains that are very different from the point of view of the trait of interest. And as you can probably see in the example that I'm talking about here, and we're going to basically, I'm going to basically explain to you some of the approaches that were used in this paper, in this, really classic paper. And I uploaded it to the Quercus website. You feel free to have a look at it. All right. So the idea is two inbred lines of tomato that differ dramatically in fruit weight, and this is just an example here. Tomatoes actually come in a gigantic variety of shapes and sizes. Everything from this giant heirloom, which gives rise to fruit up to 1 kilogram, to this tiny little cherry tomato, whose fruit weight is about an average of 1 gram. In fact, these two, if you really want to be precise about it, are two different species, but they are very closely related. The genome differs by less than 0.6%, and they will freely interbreed, okay? So you can have closely related species or strains within a species. Okay, now what do you do with these? The first thing that you're going to want to do is to cross these two inbred lines, all right? Now, I might say at this point that there are actually many different experimental designs that can be used in QTL mapping, so don't be confused if you see back crossing instead of F1 crosses. In this particular example, we're going to be looking at the F2, crossing the F1 hybrids and generating an F2 generation. There's lots of different reasons why we might want to do that. It actually gives us a chance to look at both homozygotes and heterozygotes. But in fact, the main point is that you want to take a look at the F2 generation, all right? So to map QTLs, you take your two inbred lines, right? This inbred line, what does it mean? It means that it's basically as homozygous as possible across all loci, right? As homozygous as possible. And same thing for this inbred line, as homozygous as possible, but with different alleles from parent 1, all right? So we're crossing it, we get the F1 generation, one allele from one parent, the other allele from the other, and then we cross these and we get the F2. Now note that the F2 is highly heterogeneous, And that's because, of course, we have the random effects of recombination happening, right? So, to map QTLs, what you want to do is this is the generation we're focusing on, because that's where all the genetic variation and phenotypic variation is found, right? These are all highly homogeneous, and so are these, in terms of both genetics and size, fruits phenotype. However, the F2 is where all the variation is, in terms of phenotype, in this case size, as well as genotype. So, we want to look at as many members of the F2 generation as possible, right? We want to grow these plants to maturity. We want to measure the fruit weight on each. We want to isolate the DNA, determine its genotype. And we want to do that at a set of marker loci distributed across the chromosomes. As many marker loci as we can, right? In order to find the marker loci that might be linked to our trait of interest, which is in this case, fruit size, all right? So let me just point out that there is, because that's the definition of an inbred line, that there is as little genetic variation as possible in these two parental strains. But are all of the fruits going to be exactly equal? No. And that's because of the effects of the environment, right? So there's going to be a little bit of variation due to the environment, but all of the variation is due to the environment. And that is also true of this F1 generation. They are genetically identical, even though there have been the effects of recombination, because the parent genotypes were homozygous, so recombination just results in the same genotype, right? So the genotypes are identical, so any variation in the F1 is also due just to the environment. But then here is where we have both the effects of genotype as well as on phenotype as well as the environment, okay? So what we want to do is we want to look at the phenotypes in the F2 generation and their association with genotypes, okay? So, if you remember back to my first lecture, what we actually want to do is we want to determine the frequency distribution in the F2 of the phenotype and see how it relates to genotype, okay? So, if we look at this frequency distribution again, and if you sort of remind yourself of what all these graphs that we've been looking at, right? Where here we have a trait of interest from small to large, and we can think about particular loci, each particular locus where you have one allele from one parent, this would be the small parent, one allele from the large parent, and then you have this distribution depending on whether you're homozygous for the big parent allele, homozygous for the small parent, or heterozygous and have an intermediate phenotype, all right? So what you're trying to do, in fact, our ultimate goal is to find markers where the alleles segregate in this manner where the trait of interest goes from small to large and is associated with allelic patterns like this, all right? We're trying to find markers that segregate with the phenotype and separate those out from those with no association to the phenotype. And this is by looking at the relationship between the genotypes of the markers and the phenotypes of interest. So we would not be interested in, genotype to phenotype association that looks like this, right? We really want one that looks like this instead, that sort of separates out nicely between associations of the big fruited allele with the big fruits and the small fruited allele with the small fruits, okay? Everybody's with me so far? Okay. All right. So how do we do that? We're basically doing that by using molecular markers distributed across the entire genome to genotype the individuals, and by attempting to find markers that are linked to the trait of interest, that co-segregate with the trait of interest. So how exactly do I do that? Well, this is how I do that, okay? So basically, for every single F2, I'm going to measure the fruit size, as you can see here. I'm also going to determine at each locus, and this is showing one locus, does it have alleles from the small fruited parent? Is it homozygous for those alleles? As you can see here, this is the small fruited parent, only has the T2 allele. The large fruited parent only has the T1 allele here. And then we look at all of these F2s to see what the distribution of alleles is. Is it heterozygous, right? Is it homozygous for the small fruited allele? Is it homozygous for the big fruited allele? As you can see here, the distribution of phenotypes does not match the distribution. The distribution of genotypes doesn't match the distribution of phenotypes, okay? However, in this pattern, it does match. And that's because the big fruited F2 individuals are homozygous for the big fruited allele. The small fruited F2s are homozygous for the small fruited allele, and the intermediates are heterozygous. Okay? So this is what we're looking for. This shows an association. So, this is wonderful. It's beautiful, this association. But we're biologists, and we know that it's never this clear, right? What happens if the pattern is not that clear? We need to develop a statistical method to determine if the markers are co-segregating or not, if they're associating with the trait or not. All right, so let me draw this in a slightly different way, right? In fact, it's not going to be as clear because the phenotype of interest, there's going to be variation associated with it, right? That's the first thing. Also, there might be recombination events that, again, cloud our picture. But if we plot our data like this, where fruit size is on this axis, and then here we have the genotype, if there is an association, we should see a relationship like that, right? Everybody would agree with that scenario. However, if there is no association, we're talking about a completely different locus now, right? A different marker locus, we might see something more like this, where, it doesn't really matter what the genotype is. You see this, we see a similar distribution of fruit size. So that is a clear example where there is no association. But again, so most loci we would expect will show this type of segregation pattern. However, how can we determine, because again, this is a very contrived example, right? In biology, we would see this, we would see that, we would see everything in between. How can we determine if this association, when we plot it out, is statistically significant? How are we going to determine if there is an association there or not, right? We could imagine data that's much messier. We could imagine slopes that are not as, steep. We could imagine more or less clustering of data points. So fortunately, there are statistical methods that can help us. And one of those that is in common use for exactly this kind of problem is a calculation of the logarithm odds of odds, or LOD score. This was developed to address this issue. Is there a statistical association between a marker and a trait of interest, a putative QTL, okay? So what is that? This odds ratio, right, is simply the ratio, it's called a ratio because it's a ratio of odds. It's a ratio of the probability that you see the data given that there is a linked QTL, right? Over the probability of the data given that there is no QTL, all right? Probability of linkage over probability of no linkage, all right? And then the log of odds ratio is simply the log base 10 of the odds ratio. So in other words, if the odds ratio is 100, the odds are 100 to 1 in favor of there being a QTL, all right? So in that case, the log base 10 of 100 would be 2. Okay, I'm going to do a little digression here to show how LOD scores are calculated, and this is easier to understand in the context of a pedigree, all right? So I'm going to take a little bit of a digression into a pedigree analysis of log scores. In this example, which is about neurofibromatosis, it's a dominantly inherited autosomal condition which causes nervous tissue to proliferate uncontrollably. So it forms tumorous bumps that are usually benign under the skin, but can sometimes also develop into malignant tumors. So our question in this pedigree is exactly analogous. Is this marker, is this allele, the SNP1, right, SNP, linked to the neurofibromatosis gene, or NF locus, right? Is this linked? We're going to calculate a LOD score for this. Now, you will notice this is now you're all experts at pedigrees, right? Affected individuals are in black, females in black, squares are males. Okay, so you will notice that this particular patient, right, this particular patient here inherited the disease-causing allele and the G-SNP1 from the mother. So that's this individual here who's affected, it's autosomal dominant, right? Inherited the disease-causing allele from the mother along with this SNP. He also inherited a wild-type allele at the NF locus plus a T from the father, okay? So then based on this reading, you can see that the progeny, right? The progeny inherited parental alleles, all of them, right? Some of which are not affected, which are associated with the G, affected associated with the G, right? SNP, and not affected associated with the T, SNP, from the father. However, there's one that doesn't fit this pattern because it's a recombinant, and that's this one right here. In this case, there must have been a recombination event in the father to associate the T-SNP with the disease allele, okay? So the progeny, these progeny are parental phenotypes, except for this one, which is recombinant, okay? All right. So from that, we can calculate statistics on, is this SNP locus, right, linked to the neurofibromatosis gene? And this is how we do it. Again, it's the probability of obtaining these observed results in the pedigree, right, if the loci are linked. If the SNP is linked to that disease locus, over the probability of obtaining the observed results if the loci are not linked, okay? So what we do is we take a look at the progeny. The probability of obtaining the results, right, the numerator, we assume a recombination frequency, that which is observed, which is 1/8, right? So the expected frequency of progeny would be 7/8 to the 7th, right? For these 7 parental phenotypes times the recombination probability, which is 1/8. That equals to 1/20th. The denominator, on the other hand, we assume that the two loci are unlinked. So both phenotypes are, both parental and recombinant are equally likely, right? Because we have a recombination frequency of 50%, same thing as unlinked. And so we have a half to the eighth, which is 1 over 256, okay? So that's numerator and that's denominator, all right? So it's numerator over denominator, 1/20th over 256th. That comes out to a ratio of 12.8 and a LOD score of 1.1. So the answer for this case is no. Remember, it's a log base 10, right? So about 1.1. The rule of thumb to pass the cutoff is 3. So this does not pass the cutoff for the SNP being linked to the disease site, okay? All right. So now let's go back. We know about how to calculate LOD scores. We know that we're looking for LOD scores of above 3. So in this case, where we're looking to see whether the genotypes of the markers, and the markers, remember, are distributed throughout the tomato genome, right? So if we look along a certain chromosome and we plot the LOD score across all the different marker loci on this chromosome, we're looking for LOD scores that go above our cutoff of 3, all right? And what does that mean? Why are we using this as a rule of thumb? It's because that means that it's above the cutoff that P is equal to 0.001 association due to chance. In other words, 99.9% chance that the association is real, okay? All right. Now, it is a rule of thumb. You can have situations like this where this is passing the cutoff, but that's not, right? So it is a rule of thumb, but this is what has been found to be the most useful way of defining this, all right? Now, you can always say that sometimes you don't have statistical significance because you don't have enough data. So it might be possible if you added more data that this would then become significant, all right? But at least according to the data you have now, it's not passing the threshold. Okay, so we have used these methods. We have looked through all of the markers in the genome in this cross that we did, studying with great intensity the F2 generation, right? Looking for those associations between genotypes and the phenotype of fruit size. And this is what Frary and colleagues did, right? And using these methods, they were able to hone in on a portion of chromosome 2, right? They had potential QTLs across different chromosomes. They had one very promising region of chromosome 2, and they decided to hone in on this particular region, QTL interval. Now, how big the QTL interval obviously depends on how frequent your markers are. If you only have a few markers, you don't have enough to really hone in on it, your QTL interval might be large, all right? So, now that we know, okay, there's a very promising QTL interval here with a high LOD score, how do we figure out? Okay, so we just have a region of chromosome 2. That's promising, but we really want to know what are the genes in that interval controlling fruit size. We still don't know about that. In order to know about that, in order to drill down to the level of the genes, we need to make recombinant inbred lines to help us do that, okay? And it's called fine scale mapping, and this is how we're going to do that, all right? So we create recombinant inbred lines, right, from individuals in the F2 generation, right? Each generation breeding siblings, or for plants, you can do single seed descent, inbred for multiple generations. Why do we want to do multiple generations? I mean, 8 generations, that seems like a lot. The reason is because you need them to be as homozygous as possible, right? Because recombination at each generation is created ladder-like mosaics of maternal and paternal alleles that are different in each one of the inbred lines. So you want that to be as homozygous as possible because that's what's going to allow for fine mapping of traits. Okay? So let me describe this process in a little bit more detail. So this is just the cross that I showed you at the beginning of this lecture, right? We're all familiar with this. But now what we want to do in order to do fine scale mapping is to take each one of these individuals and basically create an inbred line by inbreeding for at least 8 generations. I mean, honestly, the more the better, right? Because you really want to get rid of all heterozygosity. So, the interesting thing is that each one of these inbred lines is going to differ in where the crossover events occur. And so the distribution of maternal and paternal alleles, but they differ in the distribution of maternal and paternal alleles, but they are homozygous for those alleles across all the loci. That is the key important point, okay? And that is what allows fine mapping of traits. Everybody still with me, yes? Okay. The important thing about what I just showed here is that, remember, we have several hundred individuals in the F2, right? And they each have different associations between genotype and phenotype. They each have different genotypes because of the differences in recombination between the parental alleles that happen in the F1 generation. Okay? Everybody understands that. Recombination is random. Recombination is random, so the results of recombination would result in differences in the F2 generation. Okay? Now we're going to take advantage of that by creating inbred lines from each unique F2 individual, but by inbreeding, make them homozygous across the entire genome. So, they are all these inbred lines, that's the definition of an inbred line, right? Eliminating all heterozygosity, making sure you have homozygosity across the entire genome. However, they differ in whether or not they're homozygous at every single marker locus for parent 1 versus parent 2. And that is the important point. Okay? All right. Now, Frary and colleagues did this. They created recombinant inbred lines, each with a crossover, some with crossovers near the QTL. And this is what allowed for fine mapping of traits like this. And I will explain that process in a schematic in the next several slides, okay? All right. So this is an example, right? This is an example where you have, again, 2 tomato strains that differ in fruit weight. In this case, the example is large red Beefmaster strain, marked in red, or the small yellow Sun Gold strain, marked in yellow, okay? And what this shows is that whether the segment or the allele is derived from the large Beefmaster strain, will be marked in red at these different loci here, or if it's derived from the small Sun Gold strain, the small parent, it's marked in yellow, okay? And then these are inbred strains of the F2. And what we want to do is we can look at these inbred lines, we can measure what their average fruit weight is. Now, since all of the individuals of the inbred line are pretty much, right, have the same genotype, we can take the average of different individuals, right, to find the average fruit weight for that inbred line. Okay, we can try and get rid of the effects of the environment by doing that. Okay, So these are the different lines. And what we want to do is we want to find the locus that best matches the genotype, in other words, the allele, from the parent with the phenotype, okay? And if we look across, we can see, okay, the large beefmaster is about 180 grams. The small sun gold is about 170 grams, all right? So if we look at this locus has lines 1 through 5 all have the large fruited parental allele, but it's got a real range. Some of them have similar fruit weights as the large fruited parent, and some the small sun gold parent. So that's not a good fit. If you look across all of these, the only one that is a perfect fit is this locus here, where you can see these three have the large fruited beefmaster allele, and they're large in terms of their phenotype. These two have the small sun gold allele, and they're small phenotype, okay? So, none of the other genes are associated in such a significant fashion except for that one. And that's how they honed in on the genes that are might be responsible, okay? And then, of course, you want to be sure that you have causation and not just correlation. What I just described to you, everything is based on correlation. Correlation, there's always a small chance that you're just seeing that based on random chance. So really, the clincher is causation, all right? And so you need experimental confirmation, which is why people use model systems to confirm the effects of genes on a particular phenotype, right? So they examine the expression of the candidate gene that they found, right, using RT-PCR, and they were able to confirm that there were differences in expression of this gene. This is again in the F2 generation, in the large fruited versus the small fruited plants, all right? So everything is matching up, and now they're looking at the expression of this candidate gene, and they're finding is lower expression in large fruited plants, higher expression in small fruited plants, as you can see here, in their RT-PCR investigation, all right? And then if they look at the sequence of this predicted protein, right, they wanted to know, okay, what is this locus that we found that appears to be associated with fruit size in this experiment that we're doing? They found that its putative function was related, related to the human proto-oncogene RAS, which is involved in cell cycle control, right? And interestingly, they did not find differences in the coding region of this gene, right? Because they're looking at different alleles associated with the different parental phenotypes, right? They found there were no differences in the coding region of this gene, but there were changes in the upstream promoter region of this gene, okay? And so what they suggested is that this gene might be a negative regulator of cell division, right? Because remember, higher expression in smaller fruited plants, right? And it has a lot of structural similarity to RAS, which is involved in cell cycle control. So all of this stuff was consistent, okay, as a mechanism. So now we've gone all the way from, okay, let's, find out, let's try and figure out on a genome-wide scale which loci might be associated with changes in fruit size, all the way down to a gene with a possible mechanism for controlling fruit size in tomato. But really the clincher is you need the experiment that shows that this is actually the gene responsible, right? Right now we're still holding a smoking gun. We've got a lot of thoughts on, we've got a statistical association, we've got a lot of thoughts on how that particular gene might be regulating fruit size in tomato, right? but we don't actually have the experiment to show that it controls fruit size in tomato. And that's what makes this study published in Science such a beautiful study, and that's because they went and they actually did the experiment to show that this is the case. Okay, so they used genetic engineering to test the function of the candidate gene, right? So they took an inbred line, near isogenic line, same thing, inbred line, that was associated with the large fruit size, okay? One of the inbred lines associated with a large fruit size. But you could use genetic engineering in that inbred line. So that inbred line with the large fruit size, right, has the large fruited allele from the large fruited parent. But you could genetically engineer into that inbred line a small fruited allele from the small fruited parent, right, introduced as a transgene experimentally, and they were able to demonstrate using these experimental manipulations in the laboratory that if you do that, so you have an inbred line, the only thing you've changed about it is introducing this transgene from the small fruited parent, you decrease the fruit size in that inbred line, okay, just by the action of this one gene. So that basically clinched it. That proves that it has a role in determining fruit size, and it proves the effects of the alleles from the two different parents, okay? All right, I'm going to stop there. Thank you all for coming. If you have any questions, please post them in the discussion, and I will address them on the Tuesday second hour. Thank you.